So I'm Doug Wightman. I've been working with the PGC Alzheimer's group for 4 years and also with a website that does functional annotation called FUMA. And this is Marijn, and he also works with me as well. And he has been working on FUMA, I think, for 2 years. And he works as well with doing a project to better select causal genes from GWAS data. And we thought it'd be more fun to do it together. So you're going to see us just kind of switch in and out. Yeah, so this is a brief outline of what we're going to do. So we're going to start with a small talk about what is functional annotation and sort of what data sources do we use for functional annotations, and how can you use it to go from GWAS to follow-up studies and more biological insights into your GWAS. Then we have a small practical and a break afterwards. And after the break, we're going to talk about FUMA, which is a tool that does a lot of the things that we're going to do now just automatically for you. So it's quite a nice tool, we think. And then we're just going to wrap up. This is the general schedule, but it might be a little bit quicker depending on how fast the practicals go. Yeah. So before I start with this, just by a show of hands, how many people have had some training, maybe just a single course or class in some molecular or some cell biology? Okay, that's quite a lot. I think that's most. So that's nice, and that will make some things a little bit easier to understand. But when we're talking about functional annotation, one thing that we're very interested in is the biological interpretability of our GWAS. So there are different units of interpretation when you do a GWAS. So you find anywhere in your Manhattan plot where you have like one of these peaks, right? There's a locus where there seems to be an association with the genetics of the people that you've researched and the phenotype that you're researching. And this locus can be relatively difficult to interpret because it's just one genomic region that can be relatively large. And then what is it really doing? And even if you find the one causal SNP that might be contributing to that peak, or maybe a few causal SNPs that are underlying this locus, then still it's very difficult to know what this single SNP is doing in these people to contribute to this phenotype. So if it's a coding SNP that is within a gene, it might be a little bit easier. But most of the SNPs that we find are non-coding and are just in sort of gene deserts, and we have no clue what they're doing. Now, if we can move to gene, this is a little bit easier to go to some sort of hypothesis of what is going on. Because a gene has some very specific biomolecular function, so we can sort of start to interpret, okay, what might be going on here in this locus in cells of the people who have this genotype. And then the nicest level of interpretability, I think, is when you go to pathways or gene sets where some genes converge in like a broader biological function, and this is even easier to interpret. But going from locus to SNP and gene and pathway and gene set takes some work and also some background knowledge from the biology in these loci and of these genes. So what is functional annotation? Functional annotation basically is giving the context to these SNPs in these loci. So I sort of envision this as when you're reading a book or you're reading a paper and you just make like notes on the sides. This is basically doing the same thing but for each of the SNPs in your GWAS. So that can be whether a variant impacts gene expression, impacts protein folding, so the protein might be less efficient at doing, I don't know, some enzymatic process, for example. Maybe a variant is associated with a Mendelian disorder that sort of resembles part of a complex trait that you're interested in. And all of these annotations can sort of help us resolve these loci and find the convergence of your GWAS in this broader biological context. By the way, if you have any questions at any point, just raise your hand and let me know. I like to be sort of interrupted more than me having to gauge whether there's any burning questions. So please just let me know. So there are some very common examples of annotations, and we're going to go through most of them. But examples are so the consequences on a protein, and there's tools that can predict it. Predict deleteriousness, so we can sort of gauge how deleterious a single SNP could be to the protein structure. All of this is sort of difficult to do, and I'm not sure if you guys have been following a lot of the DeepMind stuff, but there's a new tool that now sort of accurately predicts these variant consequences for most SNPs, coding SNPs. So that's kind of interesting. That's not in here yet. It's too new. No one's really. I just saw this 2 weeks ago, but I thought it was kind of interesting. Conservation across species. If a SNP is very conserved across all species, then that might be a hint that you don't want to change it because it has like very strong effects. So that can give you a clue of whether a SNP might be important in a locus. QTLs are, I think, amazing tools for finding out what genes are affected, because a QTL is basically you run a GWAS on gene expression or protein expression, and then if the SNPs that are associated with that gene expression are also the ones that are associated in your GWAS, maybe this is the gene that is affected. So there's many types of functional annotation, and we're just going to go through some of them and visualize how these can help you reach a more biological hypothesis for your phenotype.
So the variant protein consequence is one of the more kind of simple annotations where you can say like how, where does this variant fall within relation to proteins, and will it impact kind of the amino acid that's being encoded by this? And there's lots of tools that can do it, and here's an example of ANOVAR and VEP and SNP-F, and they all do more than this, but they at minimum do this. And this might be interesting for you because your variants for your GWAS might change like a base pair, but it might not affect the amino acid at all, and that's something that's interesting to know. So then kind of a follow-up from the kind of variant annotation on that level is the predicted deleteriousness. So if you have a variant that's in a gene and you know that it's changing something about the amino acid sequence, you want to know, is that important? Because sometimes amino acids have very similar chemical properties, so it wouldn't necessarily affect the protein structure. And it can be kind of outside of the specific coding region of a protein as well. You can predict whether splicing might change, or even more granular information like the transcript information. Would it affect a specific transcript over another one? And then conservation across species is kind of visualized in this way, where you can see how conserved are nucleotides across species, with the idea that if they're in lots of species, then they probably shouldn't be changed. So if that is changed in your trait, it might be kind of information that could have an impact. And GERP is a popular tool to do that, and we'll kind of explore this a bit in the practical, but a lot of these tools incorporate lots of different information or each other, so it's not really, there's a wide array of tools you can use, and they're not necessarily independent of each other. Chromatin accessibility is a separate thing from chromatin interaction, so chromatin is kind of this mixture of protein and DNA that's how your DNA is stored in your cells, and it kind of can come in various levels of accessibility. So if it's tightly wrapped around proteins, it is less accessible to things like transcription factors or polymerases. And you can kind of assume, or they do assume, that if something is more tightly wound and less accessible, then it's probably not being read or expressed. And this is tissue specific. So you can look at your trait and find, are there regions of chromatin accessibility in your GWAS locus, and is that specific to a tissue where you would expect that to be for your phenotype? And RegulumDB is a kind of very commonly used database where you can query variants or regions, and it will annotate it with just basically how accessible this region is with a criteria, which is a little table saying are there EQTLs existing in that region? Does transcription binding sites exist? Or how accessible is it? And often based on DNA's peak, where they will take kind of a structure of the DNA and then use DNAs to try and chop it up, and the bits that are getting kind of chopped up can be seen as being basically accessible. And then QTLs is anything that's like a quantitative trait loci, so a SNP that affects something that you could measure, like gene expression or splicing or a specific transcript. And oftentimes this is tissue specific as well, so there's like another layer of kind of complication. And something really important to remember about this is, just like GWAS, there's linkage disequilibrium between these variants. So just because a variant is like the peak of your Manhattan plot and it's also an EQTL, it doesn't necessarily mean that that's the same variant driving the expression change. So there's lots of kind of follow-up like experiments and also like analyses you can do once you've found this kind of information. And then GTEx, I think, is one of the most widely used resources for gene expression data and EQTL calling. So chromatin interaction is, so the way that chromatin is kind of linearly, it's also organized in 3D space. And people assume that if two kind of regions of chromatin are more often than not close to each other, there's like some kind of functional interaction between them. And the way that the chromatin is organized is also tissue specific. And I think a really interesting example of this is the olfactory receptor genes, where lots of the chromatin is like, if you looked at the base pairs linearly, they're very distant from each other, but the way that they fold and is shaped, that lots of the genes that are relevant to, like, you know, smell and taste are all kind of close to each other and regulated by a common transcription factor. And then, so a nice thing you can do with your variants is just annotate them to see if they're disease-associated variants, and that can be kind of common traits like they have in the GWAS catalog, or it can be more clinical traits. And this plot down here is an example from something from ClinVar, where if you look at the ApoE gene, all of these little dots are a single variant in this gene, and then they have kind of, you know, experts curate to decide whether these variants are likely to be pathogenic or benign.
Yeah, so why do we do these functional annotations? There's two main challenges that we want to sort of resolve in these loci where functional annotations can help. So one challenge is the LD between variants, which can sort of obscure which variant is actually doing something. And here's a nice overview in a plot where you see the top lead SNP is this sort of like purple one, and then all of the other, you can just see how many significant SNPs are in this locus. Everything above the 8 line is technically significant, so you really have no clue which variant here is causal, since they're in so high LD. So there's a few ways you can tackle this. You can do functional annotation to sort of see if there's one variant that's maybe very clearly highly conserved and it's affecting the expression of a certain gene that you expect to be associated with the phenotype. You know, this can sort of give you some clue as to whether or not this could be the causal variant. You can try to do some statistical fine mapping, and the idea behind statistical fine mapping is, if you know the LD structure of your population, you can sort of simulate how the association signal would look like if you would just assign a variant to be the causal variant, and then you can do this for multiple causal variants in a locus and just sort of see when it converges with your observed GWAS signal. And this is nice, but I think it's been mentioned earlier today that this doesn't always work for your loci, and sadly, if your locus has very high or just very high LD, so the variants are strongly correlated, these are the loci where you really want some help in resolving it. That's also where fine mapping can become sort of difficult. And you can also do conditioning. So you condition on sort of the highest variant based on the result, and then a lot of your signal will disappear, which is sort of more of like a rudimentary fine mapping. But there's another problem, and that is that many significant GWAS variants are in non-coding regions of the genome. So even if you've done perfect fine mapping and you find that your variant is in this non-coding region, you know, what's next? What does it do? And again, functional annotations can help you here. So this is why functional annotations can sort of help you resolve what is the causal variant, but also what is the effect going to be in the biological effect of this variant going to be in the people who have this trait or disorder.
So I made these little plots to try and kind of visualize the different types of annotation and like what's happening here. So on the left you have kind of the coding SNPs, and you want to kind of see where do these SNPs exist in a gene, and if they exist in an exon, that is helpful because that's implies that it's probably going to be turned into protein, and you can see what effect that will have on the protein. And if it's in kind of introns or regions around genes, you can see would it affect splicing or expression. And then for the non-coding SNPs, you have this kind of concept that that the chromatin folds in such a way that it connects long-distance enhancers to promoters, and that's a way of stabilizing a transcription factor. You can see, does this variant that's maybe in an enhancer region affect gene expression, or is it an unknown enhancer region for a specific gene? And that's an example of kind of like what EQTL could be. And here's a specific example where it's one variant in that kind of enhancer region that's affecting transcriptor factor binding, which affects its stability to binding with the promoter, leading to decreased gene expression. And then on the right is the way that chromatin is kind of organized in these kind of loops. And you have these loops where the bits are closer together, and you might expect that that could be for a particular reason, or even with the kind of zoomed in, where these kind of loops of the chromatin are closer together, and they could be, regulated by a common factor.
Yeah, so here's another plot of a GWAS risk locus, and we can sort of see how this, how these annotations then help you resolve one of these loci. So here we have a locus with some associated variants, and there are quite a number of genes that are just situated close to these variants, and often distance between variant and gene is quite an okay predictor of whether a gene is relevant. But here are some additional functional annotations. So we see that there's 3 variants that are predicted to have very deleterious effects for the coding regions of 3 genes. We see that there's quite a lot of EQTLs that affect the expression of some genes in brain tissue. For some of these SNPs that are EQTLs, we also see that they are accessible in the chromatin. So that is an indication that these SNPs are more likely to be the actual EQTLs rather than just the LD bodies of the EQTLs in these regions. And then we see that there's one interacting region between a part of these SNPs and this gene WBB1L. And then eventually you get something like this. So you see that there's multiple sort of like variants with some annotations. And here it becomes relatively difficult, and I think this is where going into literature and trying to sort of see where this would converge with what we would expect for this phenotype, or this is where you will start to go from, okay, these are the tools that I have available, to now I have to do some research and come up with hypotheses. And basically, I always see this as just very fun puzzles to solve, and then just hope that you're right in the end. But you can do this for one locus, and then it's difficult. But if you do this for multiple loci, you can often find that there's convergence in certain pathways, which is an indication that most of these predictions are actually correct. So there's a few important things to note that we mentioned before, but I think are relevant. So there are more types of functional annotations than the one that we've mentioned here, but these are just the most common ones, and also the ones that are often in large databases that encompass the entire genome, which is nice because then you know that you're not biasing your results for just the absence of annotations for specific regions. Some annotations are tissue specific, which is often relevant if you know that you're looking at a phenotype for liver, then including kidney EQTLs might not be something you want to do. Yeah, so some annotations are sort of like the combination of multiple other annotations, and not all variants are completely annotated, and this is especially true for non-European ancestries, unfortunately. Now, if a SNP is an EQTL in a European cohort, there's a very large chance that this SNP will also be an EQTL in a non-European cohort. But the danger here, if you are looking at non-European samples, is that there are SNPs in your sample that might also be an EQTL, but weren't captured in the measuring of the European population because that SNP might just not be present there. So then you are, when you're doing your annotation, there's going to be a lot of SNPs that are said to be not an EQTL, while they might actually be an EQTL in your target population. So I think there's a definite need for expansion of sort of reference data in other populations than just European ancestry. And a lot of the biological mechanisms are unknown. So for some of these annotations, it gives you a clue that it might be relevant, but you still have no idea of what the underlying biological mechanism is. So then again, you get to do the fun puzzle of trying to figure out what that might be. Some databases are more curated, so definitely look into that, because some annotations seem nice because they give you a lot of results, but that might just be a whole lot of rubbish. And if you come up with if you find that there's a lot of evidence from multiple SNP annotations to a specific gene, you don't really know for sure if this is an actual interaction that's going on until you do some experimental work to verify this. Again, finding convergence across a lot of SNPs makes it very plausible that what you're looking at is indeed correct, but if you want to know for sure, you have to do some experimental work. Yeah, but now I say we would move on to the practical. In the Slack, we have posted a Word document where you can find the instructions. Please just go through the part for practical 1, and then we'll have a break. And after the break, we're going to talk about FUMA, and then we're going to do practical part 2. And we'll be here to answer any questions that you have, unless there are questions.
Okay, so I'm going to talk to you about the second part of the practical, and there's going to be a little bit of an explanation before we get started on practical 2. And so we're going to talk about FUMA, and if you did practical 1, you saw how tedious that experience was. It was a lot of copy and pasting, looking things up, and that wasn't even everything you could do. So now imagine doing that for all 138 loci of a big GWAS. It's not really tractable. So in 2019, Kyoko made FUMA, which is a website where the aim was to kind of aggregate lots of this reference data into a single website, and then users could submit their GWAS summary statistics and get loads of kind of annotation done without having to do it themselves, basically. So this is the general structure of 2/3 of FUMA. There's one bit that we're not going to talk about, but it's the bit that's missing from this is going from your GWAS summary statistics to specific cell types of interest. But the bit that we're going to talk about is functional annotation. So we have SNP to gene, which is kind of a module that it's its own tab on the website, and gene to func, which is another module with its own tab. And how it works basically is you upload your summary statistics, and then it does all these things. So it will annotate genomic loci for you based on kind of LD and distance. It will annotate the variants that are in those loci with lots of different information that you want. And then kind of aggregate that for you and make plots. Also, as a side part of SNP to gene, it will run Magma, which is a tool to do gene and gene set based analysis, where it will aggregate kind of the association statistics of all your variants together into genes, and then we'll give you a p-value for genes, and then the gene set just looks for enrichment in p-value in the genes in a gene set versus all the genes not in that gene set. And then the second kind of module, the gene to func, is a bit more simple. You just put in a list of genes and it will do a bunch of kind of enrichment testing for you. Yeah. So this kind of explanation of the Magma gene analysis, you just upload it, and it's the first step of the SNP to gene module. So it's just going to take all your SNPs, match them to the 1,000 genomes reference, kind of adjust for the LD between the variants, and then it kind of projects PCs based on the variants, and then aggregates those PCs to get like a p-value for each gene. And then the gene set is kind of what I said before of the enrichment of genes in the gene set versus genes not in it. And the way that it defines the gene sets is based on like a well-known kind of database called MSigDB, where they aggregate lots of different gene sets from kind of different modalities. They have kind of curated information from experts, and also kind of mining bioinformatics-based gene sets. And a nice thing as well is that you can kind of define gene sets based on tissue expression. So we have like the brain span tissue for like lots of different brain regions, expression of each gene, and then the GTEx portal is just, again, like the gene expression you look for. And that's a nice way to try and prioritize like tissues that are relevant to your disease based on the GWAS results. And then after you do the magma analysis, the next step is it will make a genomic risk locus for you. And there's 3 parts to that. They make independent significant SNPs and then lead SNPs, and then the risk locus is kind of defined around the LD of those SNPs. And I'll go into that in a bit more detail in a few minutes. Well, now actually, I guess. And then, so like step 1 is define the independent significant SNPs. So what it would do is it will sort all of the variants in each chromosome by significance. And then it will take the most significant SNP and check which of these significant SNPs are in like low or moderate LD. And if it's in low LD with the SNP that it took first, that new SNP becomes independent. And you can kind of see that with this small locus here, that the ones that are below like a threshold, and the default is like a R² of 0.6, means that these SNPs are independently significant from each other. And it will do that like repeatedly until all SNPs that are significant have been assessed whether they should be independent or not. So then the next kind of definition is a lead SNP. So again, it sorts variants from most to least significant, and then the most significant variant is the lead SNP, and then it will see take the list of the independent significant SNPs, and if those independent significant SNPs are in low LD, which is the default is R² less than 0.1, with the most significant SNP, then it becomes a lead SNP. But if they're in higher LD than this, then it's just considered to be independent significant and not a lead SNP.
Can you explain this point again?
Yeah. So like we have all these independent significant SNPs, the ones with the little arrows being pointed to them, because they're in LD less than 0.6, essentially. And then we want to know, are these like so independent from this lead SNP here, which is the most significant SNP, that it should be considered a separate kind of locus, basically, or a separate lead SNP, and it's very independent. So it's like a second threshold of 0.1. So it's just another way of defining something if it's like how independent is it from the most significant SNP.
So the R² is less than 0.6 or greater than?
So for the lead SNPs, it's less than 0.1.
For independent, it's less than 0.6.
Yeah.
But why do we see colors which are hotter than?
Sorry?
So we see colors on the markers
Yeah.
which are warmer towards the orange, right?
Mm-hmm.
Which means it's greater than 0.6?
So yeah, the on the side, the color coding is kind of explained. So like anything that's basically yellow or more red than yellow, I don't know how you describe that, is like higher than 0.5.
Higher than 0.5.
Yeah, I don't know if you can see the legend, but
So
yeah.
two SNPs are independent of each other,
Mm-hmm.
but they are not, so they have, for example, 0.2 R² each other. What does this imply in these two? What is the kind of association we are thinking about these two?
I would say that they could be independent of each other, but like it's hard to tell without doing kind of further like conditional analysis. So it's a way of saying that they're relatively independent but not completely independent. So they could be still just like a big spread of LD. Yeah, that one. The least nib is just defined based on it being the most significant one of like a certain block. But it's like not, it's more like useful to how you define loci that these definitions come from. Like the way that they are connecting to which one's causal is a bit more dubious, I would say. Yeah.
Does this also use a window-based approach to do this? In the sense it starts with a given SNP, then looks for the lead SNP in that window and then keeps moving to the next.
So at this stage, it doesn't use a window, but it does use a window for the next section. It just will sort them from most significant to least significant, and oftentimes it could be that the most significant and the second most significant are like nowhere near each other, so the LD is essentially 0, and then it becomes its own thing for the next step. Okay. So then with this kind of information that we have, we can define genomic risk loci. So another definition is a candidate SNP, and so that is any variant with a p-value less than, well, a nominal p-value, so 0.05, and is in R² higher than 0.6 with an independent significant SNP. So if you look at the figure, these blocks have been kind of defined that way. So they're everything that's in LD with an independent significant SNP and has a nominal p-value is tagged as a candidate SNP, and then the boundaries are basically the largest and smallest genomic position of the candidate SNPs. So block 1 has the lead SNP in it, and then everything that's in that boundary that I've drawn is a candidate SNP for block 1. So you've basically made these like somewhat independent blocks of SNPs. And then what happens is those blocks are merged if they're less than 250 kb apart, or the independent significant SNPs are in LD larger than 0.1. So it's like a way of further kind of spreading that block to what we would think is kind of, one locus. But like all of these thresholds are just kind of the default, and you can change them to whichever you like it to be, basically. So it gets a little bit more complicated. But kind of the idea is that if you change the threshold, it will affect the size of the window that you're interested in and the number of SNPs that are being tagged by that window, basically.
So the number of independent SNPs, the then or the blocks that we would have equal to the number of independent SNPs that we have identified in the first step.
Yeah, the blocks are used in like the process, so they're not reported out to the user. They're reported out as this final block after merging, basically. So yeah, so after all of this, you end up with a locus with these kind of boundaries, and it's based on the smallest and largest genomic position of the candidate SNPs with any of the independent significant SNPs that have been merged into this block. So it's like a way of defining a window of interest for you, and if you want it to be larger, you could maybe include like SNPs that are not nominal, or you change the R² thresholds, basically. But this is kind of important because this window is now the window of the SNPs that are going to be annotated. And FUMA does like enrichment analyses for annotations based on ANNOVA, and these are the SNPs that are being annotated. So changing this window will change kind of the results of the enrichment analysis. It will change the results of what EQTLs get tagged. So it's like something that you want to think about a bit, but you can also just work out the thresholds that make sense to you the most. Yeah, so then the next step is after all of these kind of definitions are made, you can take those candidate SNPs and then annotate them. And then you end up with like an output where it's just a lot of information in tables, which we'll see in the practical, and you can download all of that information as like text files and just interact with it however you choose. Okay, so there's now also 3 types of mapping. So you have those candidate SNPs in that block, and you want to map it to genes. And the kind of default mapping is positionally. So if any of these SNPs exist within a gene, that gene is now mapped to that locus. But you can also kind of innovate around on this as well, and you can say like, I am 100% certain that my trait like is only affected by exonic SNPs. It's going to be that, and you can choose that you don't want genes that map through intergenic variants to be included. You just only want exonic variants, and then only those genes would map to your locus. So then the second type is the EQTL mapping, where if any of the candidate SNPs are EQTLs, you can say that the gene that's being affected by that SNP, by that EQTL, is a mapped gene. But this also kind of has options for filtering as well. So you can say like a p-value threshold for the EQTL, or you have like CAD score thresholding for the positional mapping, or like regulome DB score, and like things like that. So if you have strong ideas about what would be a relevant SNP to your phenotype, you can kind of modify how genes map. And then the third type of gene mapping is the chromatin interaction. So the way I talked a bit more about how like the chromatin's organized in 3D space, we have these data sets that kind of connect two regions together. And you can say that if a region that's mapped to my candidate SNPs, like is in a way in 3D space that it's close to another gene's promoter, you can say that that gene is now mapped to that locus as well. And there's kind of two types of this where it's one way, so it always has to be your candidate SNPs are in the enhancer region, which then folds and maps to a promoter of a gene, and the high C is a different type where that kind of directionality doesn't matter. It's like both ways. If it's your SNPs in the promoter, it will also map to anything that's in the enhancer region as well. Okay, so you've done all of this and you have a locus. You get this big table of all of the loci that has been defined, and then you can click on that locus and it will make this plot for you, which we call like a regional plot, and it has a lot of this information that's been annotated for you. So at the top is just kind of like a zoomed in bit of the Manhattan plot with like a gene tract at the bottom, and you can see all of the SNPs and the LD between them, and they've been labeled if they're a lead SNP or an independent significant SNP. And you can see below the genes that have been mapped, and those are in red, and then all the other genes that are not mapped or non-coding are in blue and black. And then below that is like a separate graph, but it's linked so that all the variants are like in a line with each other. They're the same variants, and you can see the CAD score annotation, the regulome DB score, and the EQTL p-values basically for every one of those candidate SNPs. And then so there's another figure that's a way to kind of visualize this on a chromosomal level. So here is like a zoom in of a circus plot, which is of chromosome 8. And you can see on the outer edge is basically the Manhattan plot for that part of chromosome 8. And then the inside of that is the chromatin interactions and the EQTL interactions between SNPs in each locus, and the blue is a locus. So there's the blue section on the left is one locus, and the blue section on the right is a different locus. And with this one, it's interesting because the orange connections are chromatin interactions, and you can see a chromatin interaction between like independent loci, essentially, that are quite far away from each other on the chromosome. And this is kind of a nice way to visualize all of this kind of complicated information. Yeah, so then, yeah.
What about the
Green
green?
EQTLs. Okay, so that was quite a quick overview, but now we can kind of move on to the practical, where we've set up these public jobs where you can explore like GWAS results without having to submit summary statistics yourself and wait for them to get processed. So in the same Word document that you're working on before, you can just follow on for practical 2. But if you have any questions, like now, let me know. Okay, good luck. Oh, you can pick whichever trait you want. There's an option of 2. You can choose schizophrenia or neuroticism. It doesn't matter which one you pick.